Good evening, everyone. Welcome again to my YouTube channel. This is supplement tutorials, and today we'll be talking about the structure of the bacteria. In the last video, we talked about the structure of the bacteria. We talked about the major components, being it the cellular envelope and appendages and the cellular interior. And we mentioned, or we talked briefly about the cellular envelope and appendages. So we'll just go over again and then begin with the cellular interior. So generally we said that bacteria belong to the prokaryotic class of organisms, and prokaryotes have their cell nuclear materials scattered in their cytoplasm. So this is an example. This is a generalized structure of the bacteria. So we have the nucleoid, the pili, the flagellum, the cell wall, the cell membrane, and then we have the plasmids, we have the cellular inclusions. So basically these are the major parts of the bacteria. So when we pick the cell wall, as we discussed the other time, this is just a revision. We said that the cell wall is made up of what? A peptidoglycan layer, and the peptidoglycan layer is made up of two molecules that are alternating to form chains. So we have N-acetyl glucosamine and N-acetyl uramic acid, and they are cross-linked by what? Peptide linkages. So we said that these molecules, or this cell wall structure, it can distinguish the bacteria into two major divisions: the gram-negative bacteria and gram-positive bacteria. And then we said that for the gram-positive bacteria, they have thick peptidoglycan layer, but for the gram-negative bacteria, they have a very thin peptidoglycan layer. And aside that, the gram-negative bacteria have other components that also distinguish it from the gram-positive bacteria. So they have peptidoglycan layer, both have peptidoglycan layer, but the gram-negative bacteria have lipopolysaccharides. They also have porins, and they also have phospholipids. So these are also other molecules that are found within the gram-negative bacteria. In the gram-positive bacteria, they have a very thick peptidoglycan layer. They have lipoproteins, they have teichoic acid, they have lipoteichoic acid as well. So the gram-negative bacteria is known to have a very complex cell wall. So it's very difficult to destroy the gram-negative bacteria as compared to the gram-positive bacteria. They are very hard to kill. Now, aside the cell wall, we have the cellular appendages. And in the cellular appendages, we can talk about the flagellum, the fimbriae, and then the pili. So in the flagellum, we say the flagellum is basically the thread-like structure that is found around the appendages, or that is attached to the bacteria. So it's basically made up of filament, hook, and then the basal membrane. So the basal membrane is actually infused, or is actually attached directly to the plasma membrane, while the hook is the intermediary between the filament and then the basal membrane. The hook is mostly a curved substance, or a curved structure that links the filaments to the basal membrane. So the flagellum has its arrangement. So we said that a bacteria with just one flagellum attached to the end is called a monotrichous bacteria. Then we have amphitrichous bacteria. They have both flagella attached to the end. Then we have tuft of flagellum. That is the lophotrichous. That is where the bacteria have numerous flagellum attached to one end. Then we have the peritrichous. That is where it is surrounded. Alright. Then we have another cellular appendage. It's called the pili. So for the pili, we have sexual pili and ordinary pili. The sexual pili is what the bacteria uses for reproduction, and the ordinary pili is what the bacteria uses for attachment. The fimbriae also just functions like the pili. It is used for attachment. And then we have the glycocalyx. The glycocalyx consists of the capsule and then the slime layer. So they are structures that surround the bacteria apart from the cell wall. So it's just a specialization to certain specific bacteria. So for instance, when we take Mycobacterium tuberculosis, for instance, they have a glycocalyx called the mycolic acid, and that surrounds the mycolic acid is what is a slime layer. So the main difference between the capsule and then the slime layer is their texture or their structural composition. So capsules is more harder than the slime layer, as the name goes. So the slime layer is somehow fluidic and it surrounds the bacteria, but the capsule is not. It's hard, yeah, and it prevents what external damages to the bacteria. So basically, this is what we discussed today. We'll be talking about the cellular interior. Hello guys, welcome to the continuation of our video. We are going to talk about the internal structure of the bacteria or the cellular interior. So when you pick a bacteria and you begin to look deep within the internal structures of the bacteria, these are some of the components you will encounter. So first of all, you have to encounter the cytoplasm, and then you will also see the endospores, the nucleoid, the plasmids, and then the transposomes. So these structures are basic structures that are found in any kind of bacteria that you would want to encounter. Now, when we talk about the cytoplasm, just like the cytoplasm of any other cell, it is the site which harbors the other components, and it's like the overall environment of the cell. So in the bacteria, the cytoplasm appears granular, and what makes the cytoplasm granular is the ribosomes. And the rate at which the ribosomes are found in the cytoplasm determines how the bacteria is capable of replicating or dividing into other forms of bacteria. We know ribosomes are involved in what? In protein synthesis, etc. Okay. Now, the cytoplasm also contains water and other chemical substances. You know, every cell contains water and chemical substances, and they serve as a medium for chemical reaction to take place. So it's just a general concept, the cytosol. This water is called a cytosol, and it's a general concept in all other cells. Then we have the cellular inclusions. We also have certain cellular inclusions that are found within the cytoplasm, and examples of these cellular inclusions are what? Are glycogen. Okay, so they also help in chemical reactions. Okay, now we also have the endospores. So the endospores are actually the inert forms of the bacteria. You know, when we say inert forms, we mean the inactive forms of the bacteria, and it is mostly produced by the gram-positive bacteria, and basically it is in two various forms. We have the vegetative stage and the spore formation stage. So in the spore formation stage, when bacteria encounters a very arid or a very rare circumstance, or let's say a very severe condition, extreme condition, it has to present itself in the inert form, so that when it gets favorable condition, it will sprout into the vegetative form and then begin to perform its metabolic activities. Okay, so that is it for the endospores. So you have to know that the endospores are metabolically inert forms of bacteria. Then we have the nucleoids. So the nucleoids in a normal eukaryotic cell, you would say that the nucleus, but we know that prokaryotes they don't have membrane bound structure, so therefore they don't have nucleus, and they have their nuclear components scattered in their cytoplasm. So this is the same thing we have here. So the nucleus is actually made up of chromosomes that has been coiled, and we know chromosomes contains the DNA. Now, the DNA of the bacteria, or the DNA that is found within the nucleus, are also coiled and supercoiled by what we call the DNA gyrase. So the DNA gyrase is actually an enzyme that's responsible for the coiling of the DNA. Okay. Then we have plasmids. So plasmids are actually extra chromosomal pieces. So they are not part of the nucleoid. They are additional chromosomal pieces, and they perform specific and a very important function. These plasmids are mostly used in research because of the properties they confer. So mostly they code for what? Antibiotic or antimicrobial resistance. And they also code for other protective mechanism of the bacteria. So if you're able to target the plasmids, it means you succeed in what? In working against the bacteria or in destroying the bacteria. That is why this plasmid is mostly used in research. Then it has the ability to self-replicate too. So you could see everything about the plasmid tells us about how the bacteria can what? Can survive or can replicate or spread. Therefore, scientists believe that when they target this part of the bacteria, they can be able to maneuver their way into deeper research to understanding the internal or the structural components of the bacteria. Okay. Then finally we have the transposons. So the transposons are actually small pieces of DNA and they move between the nucleus and then the plasmids and they do not replicate. So unlike the plasmids, the transposons don't replicate. They don't replicate just like the plasmids. So you can see that these three structures, the nucleoid, the plasmids, and then the transposons, they are all made up of DNA, and they are all like a genetic component of the bacteria. So that is what the internal structure of bacteria is about. So all you have to know is that the cytoplasm, the endospores, the nucleoid, the plasmids, and the transposons are the basic internal structures of the bacteria. And you need not to memorize this. You just have to understand what each internal structure represents or does. Thank you very much for joining us in this session.